Thank you to the organizers for inviting me to present here, part of the recent work that I've done. And so most of what I'm going to present is joint work with Tommy Hansen, that is a master student in Denmark, University of Southern Denmark, that I've been working on, and is a paper that has been presented recently at RICOM. Okay, so what I'm going to talk mostly are mutations and cancer. Great, this works. So mutations and cancer are related because, according to recurrent theory, cancer develops starting from a normal cell that, during divisions, undergoes accumulate mutations that are changes in the DNA. And most of these mutations do not change the way the cell functions. These are represented by these black flashes here. But some of these mutations actually confer to the cell what's called a selective advantage. That means the cell can grow faster and stronger than the other cells in the body. And now we have a population of cells that are growing faster and stronger than the other ones. And these population can accumulate other mutations. Some of them are random passenger mutations. Other ones are driver mutations. And at the end of this process, we have what's a tumor. That is a group of cells that is different from all the other ones. So what we can do due to the recent advances in DNA sequencing technologies is take the cancer genome, put it in the sequencing machine, and essentially obtain, for a given patient of a specific type of cancer, all the mutations in this cancer that are all the differences between the DNA in the tumor for that patient to the normal DNA of the patient. So, of course, when we sequence and we see that this patient has certain mutations, and maybe we're only interested in the genes, so the coding portions of the DNA, we don't see which mutations are related to the disease and which ones are not. So all we see are the genes in which the patient has mutations. And to give you an idea of the numbers, if we look at one typical tumor, it will have around maybe 10 driver mutations, so the mutations that are actually related to the disease, while other hundreds or even 10,000 of mutations sometimes are passenger mutations that are not related to cancer. So since now the cost of sequencing is going down exponentially, as Dev Data showed yesterday in the plot that I'm not showing today, we can now sequence a number of cancer patients for different cancer types and obtain the mutations in all these patients. So this is the approach that has been taken by some large projects on cancer, for example, the Cancer Genome Atlas project in the US or the International Cancer Genome Consortium. And today, there are more than 15,000 samples for different cancer types for which we have measured mutations in the patient. And if we look at one cancer type, there are maybe, if we are optimistic, let's say 1,000 samples for that cancer type. Okay, so given all this data, the goal would be to identify the driver mutations, so the mutations that are related to the disease. Okay, so if I have given this talk before Ellie yesterday, now I will start to tell you that we have a lot of data, and I will really flash through all the methods that have been developed, including some of the methods that I've developed with Ellie, for which we are able to identify new important driver mutations. And unfortunately, the fact is that Ellie has already given away the secret, that is, the number of samples that we have is not very large. And so, if we were to use just black box methods, machine learning methods, without understanding what they're doing, we will not be able to make reliable predictions. So, what we need to do is to rigorously assess the significance and the quality of the solutions that we report, in particular also because what we want to do at the end is try to go to biologists and tell them that we have an hypothesis that we believe is true and that they should test in the lab. And these hypotheses are very expensive to test in the lab in terms both of time and money. So what I'm going to talk about today is then more recent works that expands from maybe what can be seen as a failure, being honest but reporting no results essentially, to identifying a new problem that maybe leads to some interesting results from the biological point of view in addition to the computational point of view. And in particular, I'm going to focus on studying mutations in association with some clinical parameter that is going to be in particular survival time. So what I mean is that for all the patients for which we have measured mutations in these patients, we also have a survival time that in essence is the time to a specific event. It could be death for the patients or recurrence for cancer or other type of events. So our goal is to identify mutations that are associated with survival. What do I mean? What I mean is that if we look now, for example, in a specific gene, we could split the patients into two groups, the ones that do not have a mutation in the gene and the ones that do have the mutation in the gene. And these two set of patients may have different survival. They may have different distribution of survival time. So let me be a bit more specific about what's the survival data that we have. So for survival, we have two pieces of information. For each patient, we have the time to the event and a censoring information. So what's censoring? Censoring is a binary variable that we can think of as being 0 if the real survival time is greater than the observed survival time. And in that case, we have a censored measure. And it is 1 if instead we have that the exact time of the event is the one that we have measured. You can think of it as if we have a 0 if we had only seen a patient up to some point and then he left the study or maybe still alive today, or we have a 1 if the patient is dead at the time that we have measured. Okay, so now how do I identify mutations associated with survival? Well, a natural way to do it is to look at one gene. For example, if we consider the first gene, that is this column, then split the patient into two groups according to the mutation status for this gene that corresponds to essentially have a vector of 0 and 1 that describe the mutation status for the patient. And then for these two groups, we can infer or approximate the survival probability given the survival time and the censoring. This is a standard procedure in statistical analysis. And then we can use a statistical test that is a procedure to compare the distribution, the survival distribution for these two populations. And what we get from this statistical test is a p-value. The t is a probability. I'm going to define it later on more formally, but essentially it tells us what's the probability to observe a difference in survival as big as the one that we see in the real data when there is no real difference in survival between the two populations. So if this probability is small, we have essentially an association between the mutation in the first gene with survival time. Okay, so this approach hinges on the statistical test that here I've given as a black box. And now let's expand a bit more on what is the statistical test that we can use. So one of the statistical tests that has been proposed in this field of survival analysis is the log-rank test. And it is one of the widest used tests for statistical data analysis, and it had been designed originally for clinical trials. So it is a non-parametric test, and we can think of it as being a function just of a censoring vector, C, that tells me for each time point if the event that I observed is censored or not, and the group label X, that tells me for the patient of a given time if it is in the first group, the one with no mutation, or the second group, the one with mutation. So these vectors are obtained after sorting the patients by increasing survival time. And so this is a non-parametric test because actually we are not then looking at the survival times, but we are only looking at these vectors, the censoring vectors, and the group label vectors. So in fact, the test statistic for the log-rank test is just a function of the vector X and the vector C. And when we focus on a specific data set that we are analyzing, the test statistics is actually just a function of the group label X. So since the censoring information is fixed, then what really matters for the gene we are looking at is just these group labels. So this is the numerator of the test statistic for the log-rank test that is given by this formula. And I'm presenting it just to show that it's not exactly an easy formula, even if it has a nice interpretation in terms of being the difference between the observed number of events in the first groups minus the expected number of events in the second group, assuming there was no difference in survival between the two populations. Okay, so now that we have these test statistics, what we can do is assume that under the null hypothesis, there is no difference in survival between the population 0 and the population 1. And under this null hypothesis, what we have is that the vector x of labels is just a random 0-1 vector of length n that contains n1 ones. So it has a specific number of ones. And now what we want to compute is the p-value, that is the probability of obtaining a value at least as extreme, so greater or equal, than the one that we see in real data using the test statistic, under this probability distribution, the one defined by the null hypothesis. Okay, so this is the test that has been widely used in the survival analysis world, and what we can do is then look at the first gene, perform the test, obtain a p-value, look at the second gene, perform a test, obtain a p-value for that gene, look at the third gene, obtain a p-value, do it for each gene in turn. At the end, we have tested many genes, for example, the genes in the human genome, and then we need to correct for multiple hypothesis correction. One way to do it is just to look essentially to do a union bound. And at the end, what we'll have is that we can identify maybe some genes that have a significant association with survival. Okay, so this is a simple procedure. Let me point out that actually, even this simple procedure requires the computation of small p-values, since we need to correct for multiple hypothesis testing. And so the standard tools that are based on the asymptotic approximation for the log-rank statistic actually do not work well. And so in previous work with Alexander Papustaki, Ben Rafael, and Eli Upfal, we have proposed a fully polynomial time approximation scheme to obtain a controlled approximation of the p-value for the log-rank test. Okay, so this is great. After obtaining this method, then we can really start to analyze the real data and see what we get. So let's say, for example, we take a data set from the Cancer Genome Atlas made of 268 samples for which we have mutations and survival data. We consider all possible genes. We perform this test. At the end, what we obtain is that there is no gene that has significant association with survival when we use this approach. So I guess according to what Eli was saying yesterday, I'm a honest statistician, or maybe I'm not a statistician. That's the easier way to say it. Okay, so we have seen no gene significantly associated with survival according to this procedure that is looking at one gene at a time. So why is that? Well, if you speak with biologists, they would say, well, looking at genes one at a time is not really the right way to proceed. Why is that? Because actually genes interact into a cell, and it is by their interactions that some functions in the cells are performed. So what we really have to look at are a group of genes that are interacting, because this group of genes are the target of mutations that are important for cancer. So for example, if this is just a cartoon of the cell, and we look at a group of genes, we have that these genes are interacting. By interacting, they perform a certain function called apoptosis. And apoptosis is a function that needs to be altered in cancer. And so there are many ways to alter this function by mutating any of these genes. So it might be that this function is associated with survival, and there may be genes associated with survival when we look at the entire set, while if we look at each gene individually, we cannot really identify this set associated with survival. So the question we want to ask then is, what are these sets of interacting genes, or pathways as they are called in biology, that are significant for cancer in general? And in particular for us, we will be looking for sets of interacting genes that are associated with survival. So what we want to do is to take the mutation data and the survival data that we had before, take a gene interaction network that describes interactions among genes, and want to identify connected subnetworks or connected subgraph that show significant association with survival. Okay, so how do we proceed? Well, first we need to define what it means for a subnetwork to be associated with survival, and this can be done quite easily. So let's say that for a given subnetwork, we have a simple subnetwork with two genes, the first gene and the second one. Now, we can split the patients into two groups, the patients that have at least one mutation in the two genes, and the patients that have no mutation in the two genes. So we're taking essentially just the OR of the two group labels defined for the two genes. And now we can use the log, we can infer the survival distribution for these two populations and use the log-rank test to obtain a p-value for this subnetwork. So to get a bit more in detail. We can use the log-rank test as before, but now instead of looking at one gene at a time, we are looking at a set S of genes that will correspond to a subnetwork, for which we have an associated group label, XS. And now we can then look at the test statistic for this subnetwork S, and we can essentially use this test statistic, or the normalized version, as a weight. So the weight for the subnetwork will be equal to the normalized test statistic for it. So this is the same slide as before, and the reason we can use this weight to, as an indicator of the association of the mutation of the subnetwork with survival is that the deviation of these, or the absolute value of this weight actually is an indicator of how large the p-value will be. Okay, so we can define more formally the computational problem that we want to solve, that is the following. Given the mutation data, survival information, this graph, we want to find the subnetwork of k genes that maximizes the absolute value of the weight for that subnetwork. So we call this problem the max connected k-set log-rank problem, and this is defined first in this recon paper with Tom Jensen. Okay, so now we have a problem that had not been studied before, and so the first thing we ask is it an easy problem or is it a difficult problem from the computational point of view? And when I give this talk to a computational biologist, they always laugh after what I'm going to show next. That is, this problem is NPR'd. They always laugh because all problems are NPR'd in bioinformatics and computational biology. Still, you need to prove it, though. So this was a nice result for us. OK, so this problem is NPR'd, but still we want to solve it in practice. So how can we solve it? Well, we have designed an algorithm that we called NOMAS, for network of mutations associated with survival, that try to identify these subnetworks associated with survival. So the way it works, the way NOMAS works, is the following. We use a color coding based algorithm to identify subnetworks of mutations that have high association with survival. And then, after identifying these subnetworks, we need to run a permutation test to make sure that since we are essentially biasing our search to identify the subnetwork that has the highest association with survival, we are not identifying a strong association just by chance. This corresponds, the permutation test essentially is what we use to correct for the multiple hypothesis testing that is implicit in our search for the best subgraph. Okay, so how many of you have seen the color coding, graph color coding algorithms before? Okay, not that many. That's good or bad, depends on the way you look at it. For me, it's good, so that the next slide is not totally ridiculous to you. So how does color coding work? Let's say that we have a graph, and we're looking for a subnetwork of size k equal to 3. So what we do is we random color the graph with the 3 colors, choosing a color uniformly at random for each of the vertices. And the reason we do this is that we can now identify efficiently the highest scoring colorful subgraph using dynamic programming. And when I say efficiently, it means efficiently when the size of the subnetwork that you are looking for is not too large. In particular, is logarithmic in the size, most logarithmic in the size of the subgraph. So the way we can identify the the highest scoring colorful subgraph, and a subgraph is colorful when it contains all the different colors that we have used, and it has cardinality exactly k. So each color used exactly 1. Is the following. We can build this dynamic programming table in which we have genes on the rows, and on the columns we have color sets, so all possible subsets of colors that we are using. And when we need to fill an entry that will correspond to identify a connected subgraph, using that in this example, consider only 2 vertices, one of color blue, one of color red, that contains the gene associated with the row. For example, this gene up here. We just need to look for the neighbors, essentially in this case, of this vertex that have a color red, since the vertex has color blue. So this can be done efficiently, and we can look at the candidate for filling up this entry, and we can score each of the candidates quite efficiently. Okay, so by random coloring that has been introduced by Alon Eusterswick in stock 94, we can identify the highest scoring colorful subgraph efficiently. And now what we have is that in this case, the highest scoring subgraph may not be colorful, but if we repeat enough time the process, we can prove that the highest scoring subgraph will be colorful at least once, so with that probability we identified. Okay, so this seems like a simple algorithm to use, so we can probably use it as it is for our method, and we just use it, modify it in not too many ways. But the main point is that when we look at color coding for our problem, for the problem that NOMAS is trying to solve, we are relying on the fact that the best colorful subgraph can be identified easily once we have colored the graph. And so if we look at this a bit more in detail, we see that this is true when the score is additive. So what do I mean that it is additive? I mean that the score or the function that defines the weight for the subgraph when I'm computing the score for a set of genes is equal to the sum of the scores for the genes. So when this is true, we have that finding the optimal colorful subgraph is easy, and so if we repeat some number of iterations of this procedure, we will find the optimal subnetwork of a certain size with probability greater or equal than a bound that we can fix. The problem for our approach is that the score we are using, the log-rank statistic, is not additive. So what that means is that even if the optimal solution that we are looking for is colorful in a subgraph, we may not be able to find it by this approach. So what should we do? Well, we may try to design another algorithm, but actually what we did is that we proved that under a model that is reasonable for mutations and survival, the color coding approach identifies the optimal solutions, and in particular identifies a subgraph that has a strong association with survival when we have enough samples. And in particular, in this model, what we have is that if we have n total genes, we have a subnetwork cardinality k associated with survival. Each of the genes in this subnetwork contributes somehow to the survival. And the other mutations that are not in the subnetwork are placed independently of survival. That seems to make sense since there are many passenger mutations in cancer that are actually not related to the disease. Okay, so under this assumption, we can actually derive a minimum number of samples that we need to obtain so that our algorithm identifies the optimal solution with guarantees essentially similar to the ones that we obtain from color coding for additive functions. Okay, so now let me show in the last few minutes some results on real data. So Tom has implemented NOMAS, and then we have tested it on different data sets from the Cancer Genome Atlas. We've used a network that contains more or less 10,000 vertices or genes, and there are 40,000 interactions. And then we run our method looking for subnetwork of cardinality up to 8 using a error probability equal to 0.01. And we have compared it to other approaches. So first we have compared it to exhaustive enumeration algorithm, in particular to compare the running time for the method. And then we have compared it to two other methods that are just general methods used in bioinformatics in particular, when one is looking for subnetworks associated with clinical data. The first one is a greedy algorithm that starts from a vertex, and that's the neighbor that gives the best improvement in the weight that defines the association with your clinical parameter. And the second algorithm is instead an algorithm that defines an additive function for the score by looking at each single gene, computing a p-value for the gene, and then looking at a set of genes that maximizes the sum of the single p-values. Or maybe we can take the sum of the minus log 10 of the p-value. Let me point out that this sum, or what you obtain here, is different from the p-value that you compute if you look at the set directly. Okay, thanks. Okay, so let's see some results. The first one is about running times. So on the x-axis, we have the size of the subnetwork that we are looking at. on the y-axis, we have the logarithm of the number of seconds that the algorithm runs for, and this is the curve for NOMAS, our algorithm. This is the curve for the exhaustive algorithm. As you can see, our algorithm is more efficient than the exhaustive one already for small values of k, and when k becomes larger, the exhaustive would not be reasonable to run. And the exhaustive algorithm is actually a enumeration algorithm for subgraphs that is not the naive algorithm that we could think of. Okay, so now let's look at the results of NOMAS compared to other approaches. And so what we have here are 3 different columns corresponding to 3 different data sets from the Cancer Genome Atlas, and 2 rows, one corresponding to look for subnetworks that have the highest association with short survival, and one looking for subnetwork that has highest association with long survival. And here we have different curves, one for our algorithm, one for different greedy strategies that one could use, and one for the additive version of an algorithm. So what we see here is that sometimes the algorithms identify solutions that are similar, but most of the time there are solutions from the other algorithms that are not comparable in terms of the weight to what is identified by NOMAS. So NOMAS identifies solutions that are better than the other ones. Okay, so now let me just show you one slide on what could be some biological significance for what we find. So this is an example of the best solution identified on this glioblastoma multiforme dataset that is a type of brain cancer. And so this is the subnetwork of 8 genes identified by NOMAS on this dataset. And when we look at these genes, we see that they correspond to parts of 2 known pathways in cancer. So one is the PI3K, AKT signaling pathway. The other one is another pathways. And both these pathways being associated with cancer before in terms of their mutations, but the group of genes has not been reported as associated with survival. And in particular, no gene in this group has significant association with survival when it's considered in isolation. Let me point out that the permutational p-value for our solution here is less than 10 to the minus 3, so it is unlikely that it's just a random effect for of chance and mutations in this dataset. Okay, so let me acknowledge in particular Tommy that has done most of the work that you've seen here, and thank you. And if you have questions, I would be happy to answer. Yep, left.
Confused. Because you said you're looking for subnetwork, whereas everything you seem to be saying was just a subset of genes. I mean, how do the edge interactions enter the picture? I mean, for example, in the planted model.
In the subnetwork, the interactions come to play into this color coding algorithm. So we are using the interaction network and finding connected subgraphs in here. So we are looking for a set of genes that form a connected subgraph. Just connected, yes. That's what we want. Yeah. I don't know if that answered your question or if it was.
Okay. If you restrict your attention to subnetworks of certain topology, like line or some simpler topologies that will make computational easier to find, and do you think that you could get also significant results, or you need to?
I don't know. Honestly, I don't know. I don't think there is a. So for some type of pathways, set of interacting genes, it's known that they should have a specific topology in the subnetwork, but in general, there is no topology that uniquely defines the set of genes that you are interested in. So I would assume you would identify some interesting sets, but not all the sets that you are interested in.
Yeah, a couple of questions.
Yep.
The first one is kind of technical. So if you use 3 colors, you can only identify graphlets with 3 vertices, or not? And if so, do you need, I don't know, 5 colors to identify?
To identify sets of size 5?
Yeah.
Well, you can identify subnetworks up to cardinality k if you use colors, if you use k colors. So, for example, if you use k equal to 8, you will identify subnetworks also of size 3, 4, 5, and so on. And actually, there is a nice paper that shows also in practice, if you want to identify subnetwork size K, you should use K+1 or K+2 colors, because that results in the optimal trade-off between the probability and the running time.
So now, for the graphs that you may encounter in these applications, what is the state of the art? Can you get up to 10, 15, I don't know?
So for color coding, for I guess in general, for applications that I've seen here in bioinformatics, you can get up to 10, and we can get up to 10 as well. For larger sets, I'm not sure that it will scale very well. There may be better ways to do it. That's another way to do it. And I have to point out again, the problem that we have defined is the first time that it is defined as finding subnetworks associated with survival.
So then the last question is, use your curiosity. I mean, have you tried to, what is the feedback from medical doctors? Did you get any?
Not regarding this paper or these results, because that's something that is work in progress. So I have to say, I showed somebody this result, the slide I've shown to you. They said it looks great. But there are more interesting results to show that will be like completely new genes, probably, that may be more of interest to medical doctors. Also have to say that there is not as much clinical data as I would like right now. There are projects that are in the plan that will generate in the next 10 years 200,000 samples of a given cancer type and all the clinical data. So I think this will become even more useful in, let's say, 5 to 10 years.
I have one question. Did you consider to use a frequent item set? Like you want something which is frequent in the sample and not frequent in the other?
That would be great, and it's a great idea if we knew at the beginning what are the two populations that we are looking at. But the point is that the two populations are defined by the subnetwork. So we don't have already the split. And actually looking for frequent item sets is something that people have tried in different scenarios, like clinical scenarios where you have case and controls, or in genome-wide association studies where you have, again, case and controls. But like in here where you have puncture patients, if you could, when you can define two groups, then you could, for example, use frequent item set mining. Yep.
Two quick questions. One, you said biologists go with K equal 10, up to K equal 10. Does it mean that there are like network with 10 nodes that have no subnetwork of size 9, let's say, or less that are relevant?
No. I hope I didn't say biologists go for K10. I said bioinformaticians, I think. Okay. So that's where you can scale with color coding.
But is there any results? Is there a result of a network of size 10 that is more associated to survival and has no subnetwork of size 9 that is?
We do have some of those.
Wow.
We do. There are no, or I shouldn't say there is no association for the subnetwork size 9. Subnetwork of size 10 has a stronger association with survival than subnetwork of size 9. Again, this is something that was not possible to do before we had this method. So is it, biologists have been looking mostly single genes, that's the answer. And when you don't see single genes, it might be that there is no signal, and that's okay, or it might be that you have to look at combinations. So what we have seems to say that there are combinations that are associated with survival that are, even when there is no single gene that is associated with survival.
Okay.